Hello everyone, and thank you for joining us today. We'll be starting now. Today's webinar, as usual, is part of the FreeMind Summer School series of webinars, and today's topic is CNS and PNS related non-dilutive funding opportunities. My name is Jonathan Adalist, and I will be giving the webinar today. Just to put it in context, this is the second to last webinar in this year's summer school. Next week, the subject will be cancer non-dilutive funding, and if that is relevant for you or interesting for you, by all means, please do register on our website or through the emails you'll be receiving or whatever. And I hope you find that webinar interesting as well. Regarding all other webinars, including the webinar today, they will all be, or actually most of them already are, on our YouTube channel. And today's webinar is no different. It's being recorded and will be on our YouTube channel with all the others. A few other, I guess, logistic issues. So first of all, if there are any questions, feel free to type them in, and I will try to address as many of them as possible towards the end of the webinar. I will have the slides available for anyone who is interested in that as well. So I'll be reaching out after the webinar, and anybody who's interested in that, I'd be more than happy to forward you the slides. And that's it. Let's begin. So a few words about who we are and who's giving the webinar today. So the FreeMind Group is a consultancy, and we help our clients raise funding from non-dilutive sources, grants, contracts, things like that. We've been around for about 16 years now. We have almost 50 full-time employees. We work with both academics, medical centers, research institutes, and obviously within the industry, very small seed stage startups and all the way up to big pharma. We submit about 350 applications a year on behalf of our clients. So we have amassed quite a bit of expertise and knowledge in this area of non-dilutive funding. The way we do that is through identifying the most relevant funding sources and funding opportunities for our clients, strategizing with our clients to maximize the application's chances of being awarded, and making it adhere to what the agencies are really looking to fund, understanding the trends, understanding what is interesting to the funding agencies and the different mechanisms, managing complex project production processes. So basically, even if it's multifaceted projects with various PIs, things like that, we manage the entire project, the entire process. We are the hub that everything goes through. We lead the joint application writing as well, and we support final contract negotiations when that is relevant. But enough about us. That's not what we're here to discuss today. Today's webinar is on the neurological space, CNS, PNS related funding opportunities. And in general, the non-dilutive funding, I guess, pocket of money is a very large source of funding, about over $50 billion a year. Within that, there are various different sources. The NIH, as an example, within the HHS, other HHS organizations like BARDA, the FDA, CDC, NSF, Department of Defense has some great mechanisms, and also there are quite a few private foundations that fund R&D activities in the life sciences. Those sources that are relevant for what we're speaking on today would probably be within the NIH, the NINDS, obviously, but also the NIMH, the NIDA, and the NIA. Within the Department of Defense, we'll be discussing some opportunities from the military and from DARPA, and private foundations, the ones we'll focus on will be the MJFF and the ADDF. So in general, and the NIH is definitely not the only source of funding in this space, but it is a very, very major one. So we'll look at the statistics for the NIH for a minute. Over $30 billion currently for this year is the NIH budget. And out of that $30+ billion, about 26 is actually going out towards research projects. So it's a very, very substantial amount of money that is available for research organizations and research projects that is available for you. And this source of funding is something that in many cases we find can be neglected, less so in academia, but much more so within the industry. And we think that's a shame, because it is definitely a source of funding that can and should be tapped. Looking a bit more specifically at the funding from the NIH in the area we're discussing today, and I'd like to prefix this by saying that these are not mutually exclusive. They're not cumulative. As an example, the amount described here that the NIH spends on brain disorders, which is $4 billion a year, and the amount that they spend on the neurodegenerative space, it's not 4 billion plus 1.8 billion, therefore 5.8 billion. There is definitely overlap there. So these are, if you add up all of these different numbers, you will not receive the total sum. There is overlap. That being said, this is a good way to look at different areas of research and understand how much funding is going there. And as you can see, I tried to take the most relevant areas out of the NIH categorical spending report. And you can see there are pretty substantial amounts of money going to a lot of different areas in the neurospace. So let's discuss in more specifics the funding opportunities, starting with the NIH. The NIH, and this is a fairly interesting topic actually, underwent a pretty major change last year, and they essentially completely changed the structure of all the mechanisms that they use to fund R&D activities. If you search for grants in most areas, you'll find R01s, R21s, things like that, standard SBIRs. And in this space, it will be fairly different, and that is because in 2014, early 2014, they call it a paradigm shift. I don't know if that really is deserved, but they definitely changed it very drastically, the way they fund. Essentially, they divided the entire scope of funding to 3 different areas: biological, small molecule or chemical pharma, and medical devices. Each one of these areas has their own mechanisms, and we'll be discussing in much more detail each one of those in a few minutes. But generally speaking, this is the first division they made. They divided it into these 3 main categories. And then within each category, let's just go one by one. In the biological area, they have the Create Bio program, which is divided into the discovery track and the development track. And we'll look at these more specifically in a minute. But generally speaking, the discovery track is for more preliminary research, for discovery, as the name does kind of give you a clue. And the development track is for more advanced, developing the leads, research, even towards the clinic as well, and the clinic itself. In the chemical pharma area, it's the BPN, the Blueprint Neurotherapeutics Network, and this covers a pretty wide spectrum of stages of development. Again, we'll touch on this more specifically in a minute, but generally speaking, from discovery and all the way through the clinic. And within the devices, there are 3 different tracks: Final Design Program, Path to 510K, and Path to PMA or HDE. Again, we will be discussing these soon. And basically, I failed to mention this, but both Create Bio and Create Device are create mechanisms. Create is for cooperative research to enable and advance translational enterprises. That is how they named it. And let's look at them. So first of all, before we do that, I'd like to kind of make a distinction. The mechanisms they switch to, the mechanisms we'll be discussing, are U mechanisms. And I gave a pretty comprehensive webinar specifically on the subject of U mechanisms 2 weeks ago, and as I mentioned earlier, that webinar is available on our YouTube channel. So if you want a little bit more insight as to the specifics of what a U mechanism is, then by all means, please do check that out on our YouTube channel. But in a few words, I can say that essentially the U series of mechanisms are cooperative agreements. And basically, these are used when there will be substantial federal scientific or programmatic involvement. This does not mean that they will make decisions for you, but the NIH will want to be part of the process, understand the decisions, be part of the discussion. Basically, they feel that way. They have more understanding of what's happening, and they can, although not control, at least influence to a certain extent what happens with the research and the funding that they give you. Many CEOs, CFOs can be alarmed by this initially, but I can say across the board, that from our experience in the past, clients that have been awarded with these mechanisms find the NIH's input to be very valuable. So I have definitely advised not to be deterred by this. It's actually, as far as we are concerned, it seems like it's a very positive thing, actually. The projects are milestone driven, and there are SBIR versions for pretty much each one of the mechanisms that we'll be touching on, and you'll see that. But basically, it's an important distinction because basically all of these mechanisms will have, well, actually not mechanisms, but tracks will have more than one mechanism, and you can make a decision as to what the best one for you is. Essentially making sure that almost any research direction or idea or project within the neurosciences will have the right mechanism here, which is a great thing. Basically, I think the bottom line, the take-home message here is no matter what you're doing in the neurosciences, there are grants for you, bar none. And that is a great message. That is a pretty tremendous thing to say. And I can say that with a very fair amount of certainty. Looking a bit more specifically at the mechanisms themselves that this paradigm shift, so to speak, utilizes. So there are the UH2s, the UH3s, the UO1s, and the U44s. UH2s and UH3s are not SBIRs, but they are similar to the SBIRs in the essence that they are phased. You will not submit usually a UH2 separately or a UH3 separately. You'll submit a UH2, UH3. And upon completion, you'll submit it as one application, not two separate applications. Upon completion of the UH2 phase, you will start receiving the funding for the UH3 phase. A U1 is essentially the cooperative equivalent of an RO1, so it's a distinct project that will be funded from A to Z by the funding agency. The main difference between the UO1 and the RO1 is the fact that it's a cooperative agreement. So, as I mentioned, the program officer will be part of the discussion. They'll have a better understanding of what you're doing. Also, very, very important, and again, this is something I emphasized many, many times in my webinar 2 weeks ago on the U mechanisms, but I'll say this once at least this week because we will be discussing so many U mechanisms. Very important in collaborative agreements and U mechanisms to be in contact with the program officer before you submit. Do not make the first time they receive the application or they look at the application when they're actually reviewing it or deciding if they want to give you funding. That is generally a very bad idea. And unlike program officers and other mechanisms, they will be open to discussing it with you beforehand. So absolutely utilize that. And the U44s are SBIRs. It's a collaborative SBIR, and it's a phase 2. Actually, there are pretty much two options in a U44, and that are not, both of them are always possible, but generally speaking, U44 can either be a fast track, meaning a phase 1 and a phase 2, just like I mentioned for the UH2, UH3, or a direct to phase 2. Sometimes both are optional and you choose one, and sometimes it will be specifically one of them. So, a bit more in detail. The UO1 budget is up to 4 years, $500,000 a year, plus 40% indirect costs, so it's up to about $2.8 million of budget. And as I mentioned, the period is up to 4 years of funding. UH2, UH3s have, and this is also including the indirect cost, usually that's 30 to 40%. We calculated it here at 40%. So up to 2.8 million for the phase, the UH2 phase, and up to 8.4 million for the UH3 phase, where the UH2 phase can be up to 2 years, the UH3 phase can be up to 4.5 years, but the total period of the grant that the funding you're asking for cannot be over 5 years in total. So you have to decide how you want to divide that. A U44, the SBIR, for the phase 1 is $500,000, and for the phase 2 is $2 million. And the period is up to 1 year for the phase 1 and up to 3 years for the phase 2. All of these mechanisms will have their next deadline February 11th of next year. That may sound like it's more than a few months away, but like I mentioned, these are fairly elaborate grants, all of them, some of them more than others, but generally speaking, they're not small grants. And it's very important to have a discussion, or rather discussions, with the program officers in advance and have their involvement and get their feedback and tweak your application to what they're really looking for. So I would definitely recommend, if not starting to work on it at the moment, at least kind of directing yourself in the right direction and reaching out to the program officers. You definitely cannot do that too soon. So, discussing a bit more specifically the Create Bio first, these projects. So the first track we'll discuss is the discovery track, and as the name suggests, it's for earlier research discovery, optimization and preparation for development of biotechnology products and biologics. That is the name of the solicitation, and there is a U01 version and a U44 version. U01, as I mentioned, is a research project grant, a collaborative research project grant, and a U44 is an SBIR. So there are different mechanisms, and you need to decide which one of those is the right fit for you. In many cases, if you are SBIR eligible, the SBIR version may be the right choice, but again, this is something that really needs to be discussed with the program officer and understood in depth. The U01 budget is up to 2.8 million. The SBIR budget is up to 2.5 million. In the development track, or rather the preclinical and early phase clinical development for biotechnology products and biologics, as the name suggests, it's for more advanced, not for discovery, but for preclinical and early phase clinical development. There are, again, two mechanisms available, the UH2, UH3, and the SBIR version. The UH2, UH3 has a budget that can be actually up to 10 million if you combine the two phases, and the SBIR has a budget of up to 2.5 million. Something that's worth mentioning, some mechanisms have no budget cap, and you can, although you have to justify what you ask for, you can ask for as much as you want. That being said, it is a real rarity to have a recurring solicitation with a defined budget that is this high. These solicitations have two deadlines every year. They're recurring. The next one is February, and after that will be August, and the next year again February and August. So these are not rarities. They're not one-time things. And to have something like that with such a high budget, and that is part of the defined budget. This is not something you need pre-approval for. This is not something you really need to work. Well, obviously you always need to justify what you ask for, but it's not like asking for 10 million from an open solicitation that is expecting a $2 million budget. Although technically possible, you really need to work to justify that, and they really need to love your ideas in order to agree to that. Here, you're talking about a solicitation that, as per their definitions, may be willing to fund you with up to $10 million with two deadlines every year. That is something to consider very seriously. The next area we'll discuss is the small molecules, so the BPN, and the specific mechanism here, or rather the specific track, is small molecule drug discovery and development for disorders of the nervous system. So basically, this is for both discovery and development. So it as I mentioned earlier, will pick you up from discovery stages and take you all the way to the clinic. Not necessarily, depending on the project period, sometimes it won't fund you all the way, but you could submit this regardless of where you are. So it's open to all of those stages of development. If you can cover all of that ground within the time period that they're willing to fund, then they may take you all the way. But regardless, it's open to all those stages of development. Two versions as well, two mechanisms. The U44, the SBIR, has a budget of up to 4.4 million. And the UH2, UH3 has a budget of up to 3.5 million. Next deadline, again, as I mentioned, February 11th. The last area of the three general areas that they divided it into is the medical devices, and that would be under the Create Therapeutic Devices program. That is divided into three. The first one of the three is the Final Design Program, Translational and Clinical Studies to Inform Final Device Design. Two mechanisms here, and this is basically for medical devices that need a fair amount of experiments, trials, whatever it may be, in order to be ready to submit to the FDA. Not something that is close to being finalized. And if that's the stage that you're at, then this is probably the right mechanism. The UH2, UH3 version is up to 10 million again, and the SBIR, the U44, up to 6.5 million. Very high numbers here. 6.5 million for an SBIR is extraordinary. If you are a bit more advanced than that, then essentially choosing between the following two is based on the regulatory path that you expect or know your device is going to follow, either a 510K or not a 510K. If you're going in the 510K path, then the right mechanism to fund your activities is the translational and clinical studies on the path to 510K. And that is either a UH2, UH3, or U44 again, same sums as earlier. And the same thing is true for the path to PMA or HDE. Essentially, the only difference, as I mentioned, is the regulatory path that you are going to follow. Both have the same options for mechanisms, and both have the same budgets and time frames, as well as deadlines. This is an interesting subject, and all the changes I've been discussing up until now occurred early 2014, so there have already been a few deadlines. It's relatively new, but it's not really brand new. This is something that's even newer than that. Essentially, the NINDS, this is late 2014, realized that there is a gap in the early discovery phase. There are a lot of projects that are interesting to them, they would be interested to fund, but that are not quite ready for the criteria they set for either the Create Bio or the BPN programs. What they did is they issued this in December of 2014, and they're called the Ignite, so it's innovation grants to nurture initial translational efforts. Essentially, it's a new series of mechanisms designed to segue early stage projects into the Create or BPN programs that we discussed earlier. So this is really for early stage research, and this is not necessarily, well, not necessarily, it's not relevant for medical devices. As you can imagine, it's hard to think of what discovery stage medical devices would be, but regardless if it is a small molecule or a biologic, this is definitely relevant, and there are 4 programs in total that will be introduced. 2 of them have been introduced, and they were introduced, as I mentioned, in December of last year, and 2 others that are expected to be introduced very shortly, actually. First 2 here, the assay development and PD and efficacy Ignite programs are currently available, and the 2 following ones, the animal model development and the platform technology development mechanisms, are yet to be issued, but they are expected to be issued very shortly. Excuse me. So let's look at the 2 that have been issued. So this is for assay development, and if you are looking to develop an assay for screening efforts, excuse me, that would be relevant for screening for therapeutics for neurological disorders, then this is a grant that could fund you up to $750,000 to develop the assay. So this is for very early research, not even screening. It's to develop the assay to conduct the screening. Very interesting that they have mechanisms that go this early, and definitely worth utilizing if this is where you are. Next deadline, and this will be true for a few of them, is October 16th. This is in the very immediate future, so if you are in the right area to consider this, I would definitely recommend reaching out immediately, although this is not a U mechanism. Notice that this is an R21, R33, so essentially it's different in that essence, and you don't need to have the program officers as involved, but still, there's not much time until October 16th, and you definitely need to get going. Excuse me. The second one that has already been issued is pharmacodynamics and in vivo efficacy studies for small molecules and biologics or biotechnology products, also in R21, R33. By the way, I failed to mention this discussing the previous solicitation, but R21, R33s are essentially also phased mechanisms where you receive the R21 funding initially. Upon completing the R21 phase, you receive the R33 phase. You submit it all together as one application. You do not submit 2 separate applications. In this case, they basically fund the pharmacodynamics, pharmacokinetics, and in vivo efficacy studies on your molecule. Excuse me. So definitely also worth utilizing. Also a budget of $750,000 and the same deadline, which is approaching very rapidly. The 2 new mechanisms that are expected to actually, initially we expected them to already be issued at this point, but the NIH themselves said they expect to introduce them mid-2015, so it really should be very, very shortly. I don't know if it'll be issued in time for the October deadline, maybe not. It may be, the first deadline may be in February, but still, regardless if they release these in time for the October deadline or not, these are two programs that are definitely worth looking into. The first one is for development of animal models, and the second one is for development of platform technologies. Very interesting. They will actually fund stuff, projects as early as trying to develop an animal model to check your molecule, or trying to develop platform technology that will allow you to develop therapeutics for diseases. This is very early research, and something that is not usually funded this easily. Not to say it's easily, it's probably going to be fairly competitive when it comes out, but it's definitely something that is very valuable to know about. A few other opportunities in this space, and I won't go into a lot of detail here. These are a little bit, they're not as new. Some of them, such as the drug discovery for nervous system disorders, we also do not expect to be continued after it is expired late this year, because it kind of overlaps pretty drastically with some of the IGNITE programs and the CREATE and BPN programs. But essentially, these programs are paired solicitations, so there is an R21 phase and an R01 phase. These are not phased in the essence that you cannot submit them separately and you go one by one, but there is just in the same subject an R21 that you could submit or an R01 that you could submit. You essentially decide based on the stage of development you are at if you're going to submit an R01 or an R21. And some additional sources of funding that are worth looking into, especially if you're ready for clinical trials or in the clinic, are these opportunities. And it's interesting to see an area of research, in this case neurosciences, that has so many different solicitations and opportunities to support clinical trials. That's not always the case, and if this is where you are, definitely worth taking advantage of. Another source of funding that funds activities in the neurology space is the Department of Defense. The first solicitation I would like to discuss is the USA MRMC BAA. Basically, this is the general mechanism that the military uses to fund essentially all of the different projects it funds. There are a lot of different programs within this solicitation. The relevant one here is the CCCRP, the Combat Casualty Care Research Program. That one of the areas of focus there is research and development of technologies to diagnose and to limit the immediate short and long-term impairments that follow TBI and spinal cord injury. So if this is the area you're in, this is a great mechanism to use, especially since there's no budget cap. If they really like your ideas, they will fund you. Definitely worth utilizing. Second BAA opportunity I'd like to discuss is a BAA from DARPA. Important to mention here when talking about DARPA, DARPA only fund really cutting-edge, extremely innovative ideas. Something that's just a little bit better than something else that's out there is not interesting to them. They look for things that will change everything. If you have an idea that really is groundbreaking in this area of research, then one of the focus areas for this BAA that they have currently out is discovering and leveraging novel insights from neuroscience, psychology, cognitive science, and related disciplines to advance treatment and resilience in neurological health and optimize human aptitude and performance. So if you are in this area, and as I mentioned, you have something that truly is groundbreaking, definitely consider submitting to DARPA. No budget cap again, and that can be fairly attractive. This and the previous one are both rolling BAAs, so there's no specific deadline. You can, until the program closes, you can submit whenever you're ready. And depending on when you submit, it will be reviewed, and you hopefully will be asked to submit a full application, because initially you submit a pre-application. That's how both of these mechanisms work, so it's a good thing to use in between other grants since it's open year-round. Private foundations are also a good source of funding in this space. Start with the Michael J. Fox Foundation, and this is obviously in the Parkinson's disease area. They have also actually recently changed their funding programs. They currently have 3 general programs. Each one of them has a few mechanisms within it. It's targeted advancement, therapeutic development, and outcome measures. Before I go into the specifics, it's worth mentioning that they will be holding a 45-minute informational conference call with anybody interested in learning more about the specifics of these mechanisms. That will be on August 19th. And if this is something that could be interesting to you, I definitely recommend attending that. You would need to RSVP on their website to be able to attend. But again, definitely worth it if this is something you're considering. It's free, obviously. So going into a bit more detail, the 3 programs are, the first one is Target Advancement Program, and this includes 2 mechanisms. One is for Target Validation Awards, and the other one is Target Optimization Awards, and I think they're pretty self-explanatory, either validation or for optimization. In the Therapeutic Pipeline Program, the mechanisms are disease-modifying strategies and symptomatic strategies. And in the Improved Biomarkers and Clinical Outcome Measures Program, you have the option for imaging studies, and clinical or physiological studies, and biochemical assays and outcome measures. All of these are mechanisms you could use depending on the area of science and the project you're suggesting. Another source of funding in this space is the Alzheimer's Drug Discovery Foundation, the ADDF. They focus on drug discovery and preclinical development, early detection, clinical trials, and prevention, all 4 of those areas. They don't fund basic research, excuse me, and they're looking for novel targets and therapeutic approaches for Alzheimer's disease and also related dementias and cognitive aging. So again, both this and Parkinson's disease, basically Michael J. Fox, are great fits for specific areas within the neurosciences, and if that is your area, then by all means utilize these as well. Actually, because it's not part of the NIH or the DoD, you could submit this almost the same application covering the exact same activities to both, and then if you win one of them, hopefully even win both, then if you win both, you have to decide which one you're going to collect on. But a big advantage of having it from a completely different source is you can submit the same projects to more than one, and that way you significantly increase your chances of being awarded. So those are the main solicitations I wanted to cover today. I think it's worth touching on how the review process works, and this is an example from the NIH website, but essentially this is true for all sources. Essentially, it's a risk management process, the review. They're weighing the risks against the strength of your proposed project. There are 5 criteria that it will be judged on: leadership, environment, significance, innovation, and scientific approach. And basically, they'll be asking the questions: do you have the right leadership in place to helm this project? Do you have the right environment? Do you have the lab space? Do you have the necessary equipment? Is this really significant? Is this project that you're proposing really going to make a significant difference? Is it innovative? Is this something that is really new, something that has not been done? And all the answers to those questions have to be yes. But really what makes an award either yours or not will be your scientific approach. You need a top-notch scientific approach in order to be awarded. All of that is weighed against the risks, and knowing how to convey the strengths of your project in a way that makes them know that there are significant benefits from this research, but also that you know how to deal with any unwanted results or events is also part of making sure that you're presenting the best possible application that reduces the risk component and therefore increases your chances of being awarded. We advocate a systematic approach. You have to understand the interests of the agency as well as you possibly can. You have to focus your application to those interests. You have to ask for what is necessary. Don't ask for too much. Don't ask for too little either. Ask for really what is necessary. You have to present a complete project, cover all the different facets, everything that needs to be addressed should be addressed. All the different capabilities that you need to have, you have to present the necessary people, the necessary equipment with those capabilities. If you don't have a certain set of capabilities, if there's a gap in your capabilities, then by all means collaborate. Find the right person who is capable of carrying that out in the best possible way. Collaborate with them. Not necessary. There's no requirement to collaborate, but there is definitely a requirement to cover all bases. And if you do not have all bases covered, then by all means, definitely collaborate. And if you do collaborate, try to find the best possible collaborators you can, obviously. Target the right mechanism. There are a lot of different pockets of money out there. You have to find the right one for you, and I think a lot of what we discussed today is good direction in that way. I think also the changes that they made make it a lot easier to do that in this space, and that's a very positive thing, I think. Different sizes of awards and success rates with the different mechanisms, and we touched on that. It's very important to understand that when strategizing as well. You have to conduct a thorough strategic assessment, understanding all of these things, and planning where you're going to submit and when you're going to submit the applications. And basically that leads into what we call a multi-submission granting strategy, that is basically laying out all the different opportunities and deciding which ones to write, when to write them, how to write them, when to submit them, and all of that. That we find is really a key in being successful at raising non-dilutive funding. The FreeMind team, not the NIH team, as I mentioned, almost 50 full-time employees, a smaller team of analysts, a larger team of project managers and writers. I'd like to thank Dr. Ina Gorodovsky, one of our senior analysts, that helped me put together this presentation today. And generally touching on the service we give and how we do what we do. Essentially, and I think I pretty much laid this out, but being a bit more accurate in it, it's two, I guess, separate parts that are essentially one whole, the strategic side and the project production side. So strategizing, understanding what the best sources are and how to approach them, and then submitting application by application by application, and always updating the strategy, understanding what has changed, adding new sources, adding new applications, incorporating them in the plan, producing those applications as well, and that's essentially the process. Starting with the R&D funding needs of the client, creating a long-term strategy, based on that, creating a strategy for the next few months, submitting the necessary applications, getting them reviewed, hopefully being awarded, and again, essentially that's how we do it. At this point, I will answer some questions. So let's see what we have here. So there's a question here about collecting from both the NIH and non-NIH sources. I think I touched on that, but just to be sure, you cannot collect on the same project from two sources. That's actually considered fraud. But you can definitely submit to more than one source, and that way maximizing your chances of being awarded. You can also divide the same project into sub-projects and submit them separately, and that way you can also maximize your chances. But you cannot fund the exact same set of activities more than once. Here we have questions, and absolutely. So, this is a fantastic question, actually, asking about CDMRPs. There are relevant CDMRPs, and thank you for asking this question. I actually intended on saying something about the CDMRPs, and I ended up not saying it, but since you asked, I will touch on this. There are relevant CDMRPs, most, I guess, prominently for TBIs, spinal cord injuries, things like that. Definitely, and by the way, the CDMRPs are congressionally directed medical research programs. The reason I didn't add them after a discussion with our analysts was basically because the next deadline we expect to be in almost a year from now. So not necessarily relevant right now, but yes, CDMRPs could definitely be an additional source of funding in these areas. Let me see if I can find another question here. There's a question about non-NINDS institutes within the NIH and their interests in funding SBIR, STTRs. Absolutely. obviously depending on the science. But all the sources I mentioned earlier, be it mental health, alcohol abuse, aging, depending on the specific project, if it's in their scope, and in many cases it is, then they are more than happy to participate in the funding. And actually, you can see in the different mechanisms which of these agencies are on board, and in many cases that is exactly what happens. I can say that if you look at the about $5.8 billion that are currently going to the neurosciences, almost half of that is not NINDS. So I guess that's the best way to answer your question. Let me see. Okay, there's a question here about non-US companies. That's a fantastic question. I guess I'll start by saying yes, you're definitely eligible for this type of funding. Basically, you would obviously not be eligible for the SBIR mechanisms. What you would probably do is in each one of the different areas that I laid out, be it the BPN, be it the create bio, be it the create device, and regardless of the stage of development, you would go not for the SBIR version, not the U44, you would go for the UH2, UH3, or the U01, depending on the mechanism. And those are typically open to non-US entities. So to answer your question, yes, non-US organizations are definitely eligible for this type of funding. Another question here about the recording. I've seen a few of these, so I guess I'll repeat it. Maybe not everybody was here initially when I mentioned this. So the recording will be available on our YouTube channel, and I'll be sending you all a link to that. That's it. I would like to thank you very much for attending today. And please feel free to contact me with any additional questions you may have. And I hope you register and enjoy our webinar next week. Thank you all.